All right, let's do it. Let's do this. Um, we are going to do a GFP Transformation Lab. So let's start the lecture. So now, um, before I start the actual theory behind it, let me just review a little bit about some bio stuff, intro bio stuff. If you remember, you have two bacteria with two different traits. If you remember from general bio, you had, the process, central dogma scheme, what you guys call it, DNA makes an RNA, makes a protein. The process, DNA is transcribed into messenger RNA. mRNA is translated into polypeptides, and that falls into a protein of some sort. And then that, causes the microbes at the end to organisms to have two different traits in this case, so affects the phenotype, right? So, keep that in mind. The E. coli, or in this case, prokaryotes in general, don't carry a whole lot of proteins because they are pretty simple organisms and they don't really express things that's not necessary. So compared to eukaryotes, they're pretty simple, but so so keep that in mind. But anyways, going back, what makes E. coli and E. coli? Let's see. So if you look at right here and right here, so say, this part right here, so what makes an E. coli and E. coli is a nucleoids. Nucleoidal chromosomes, they're all the same among all E. coli, but the particular different traits come from different plasmids. So what makes an E. coli and E. coli is nucleoids, but here, pictorially speaking, there is a black circular DNA plasmid here, red circular DNA, another plasmid. So, so let's look at these two. So, in general, in bacteria can exchange traits by conjugation process. So, this plasmid can go into the other bacteria, and hence this E. coli, let's say, can pick up the particular trait that used to be in this, or that is the same trait as this particular E. coli. Anyways, but we are not doing that. We are not doing conjugation. We are actually doing transformation. So we are actually inserting the plasmids into the E. coli, and then that has particular traits. In this case, the trait is um, green, fluorescing green. So it's a green when you have light, UV light being applied, that is absorbs the UV light and then fluoresces green light. So the phenotype in this case is green fluorescing E. coli, except that what makes fluorescing green is the real question, right? So that's the protein that fluoresces green. Now, GFP, green fluorescing protein, comes from jellyfish. We're going to talk about that in a minute. But basically, that particular protein is inserted into or was inserted into pGLO vector. So you are provided with pGLO vector plasmid, and basically we're going to transform E. coli, the strain HB101 strain of E. coli, which is non-pathogenic, non-infectious. It's pretty safe uh, E. coli. But we are going to transform this E. coli with a pGLO vector, and express GFP proteins. And also, pGLO vector also contains a couple of other genes, important genes that you guys are dealing with. It's called, the region's called RC, PBAD, and GFP, of course, and BLA gene, and ORI. ORI is just the origin. It's the you can consider it sort of like the reference place, but that's where the replication start taking place. It's a circular DNA, so you kind of have to have where the replication starts, and that's ORI part, but we don't worry about that. Let's see, we have RC region, PBAT region, GFP region, and blah region. Anyways, just to for the pictorial representation, there's a square side and arrow side. This pointy side, so square side to pointy side, that's how the expression takes place. So keep that in mind. There's a PBAT is a promoter region for the region that it used to be a coding for an era B, an era A, and era D proteins, which are catabolic proteins. And then BLA gene encodes for a beta-lactamase, which is an antibiotic hydrolyzing enzyme. We're going to talk about that in a minute. So again, so the important part, one of the important parts is a PBAT region. That's the promoter region, which was attached right before the GFP region, because the GFP was derived from or isolated originally. This particular gene was isolated from jellyfish's gene. So it's a jellyfish, obviously it's a eukaryote. So the prokaryotic gene, when you actually recombinantly inserting a GFP, and if you just brute force inserting and hope for the best for expression, doesn't generally happen because again, the mechanism for expression is slightly different. So in this case, promoter region was also included right before GFP, so that the promoter region allows the initiation of the RNA polymerase to start expressing and then going along, and then the GFP, and then the expression continues. So PBAT region was inserted just so that the expression of GFP becomes a little bit more possible. So keep that in mind. Again, it was recombinantly inserted restriction enzymes and stuff, which you will be probably talking about now or soonish. Anyways, also BLA gene, this is the region that provides the antibiotic resistance because it encodes for a protein, an enzyme that hydrolyzes beta-lactam. Beta-lactam right here, this is an active ingredient for beta-lactam. There's an amide 4-membered ring, but this particular region provides, again, the encodes for a beta-lactamase, which is a enzyme, right? Enzyme that hydrolyze the beta-lactam. It actually hydrolyze the amide linkage right here, and then hence inactivating the beta-lactam, which is it's an active ingredient for a penicillin type drug. So just so that you know, right? For our case, we are using ampicillin. It's a penicillin derivative. And then, so if pGLO is expressed, a beta, BLA gene is also expressed, and hence it provides antibiotic resistance. So we can we can tell if the transformation took place or not by looking at the expression of GFP, but actually by looking at the antibiotic resistance. And also you can selectively keep the antibiotic resistance because of the transformation and inclusion of the antibiotic resistance through a BLA gene. So keep that in mind. So now, again, let me emphasize, if pGLO is expressed, if the transformation is successful, and then it'll survive. So keep that in mind, all right? Your colony of E. coli will survive, right? Whether you have GFP or not is a whole different ballgame, because there's a expression of GFP is regulated by the presence of arabinose because of the RC protein. So let me talk a little bit about RC protein and then PBAT region. So RC, RB, and RA and RD are the region that's PBAD. This bad comes from this region called area B and area A and area D region, which encodes for a catabolic proteins or catabolic enzymes for arabinose. The arabinose is a six-membered ring sugar, so it's just like glucose. Um, so it can be used as an energy source. Of course, when just like I said earlier, the 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 prokaryotes don't produce these proteins unless needed. Like for example, if the arabinose, or if the energy source isn't there, there's no reason for them to expend energy to produce these proteins. So it'll just be inactivated and it just doesn't produce these proteins. So again, the presence of arabinose sugar, if it's there, then you want to be able to use arabinose as an energy source, and then it expresses these three proteins that has something to do with the arabinose catabolism, breaking down and using it as an energy source. So keep that in mind. Now, so let's say in wild type E. coli, you have ARA-C gene, and that encodes for ARA-C protein and sits right here in this region. So imagine, so in this case, like, so like, what I just said earlier, you don't want to express unnecessary proteins if you don't have the energy source. So expression in this case, ARA-C protein sits right here. So RNA polymerase can't really express the genes transcribe these three proteins. So now, think of it pictorially speaking, just conceptually speaking, just think of it as ARC protein sits right there and just prevents the RNA polymerase to latch on and start polymerizing. Now, so again, if you don't have arabinose, no expression. Now, imagine you have arabinose now, and then this arabinose binds with ARC protein, or ARC protein binds with arabinose, but when it does, it changes the conformation. So, ARC protein now changes its conformation by binding with arabinose, and then it sort of like moves out of its way and allows the expression of these three genes so that you can utilize arabinose in the organisms to be processed for an energy. So keep that in mind. In this case, messenger RNA can start, RNA polymerase can start transcribing these three regions. Now, imagine that this particular region, again, promoter region is still there, so that the messenger RNA or RNA polymerase can start expressing whatever the subsequent genes. But anyways, so you have PVAD region, but you have arabinose, and imagine that the ARAS B and ARA A and ARA D region was replaced with GFP. So in this case, you have arabinose. Again, the ARA-C protein, it's still, again, it's clinging on, but it moves out of its way by changing its conformation. So going from here to here, and then expression can start taking place. All right. So, again, pictorially speaking, that's how the expression GFP gene is read, and then translated into GFP proteins. Now, so again, so think of arabinose as an on-off switch. So if arabinose is there, expression takes place, but that's only because ARC protein is there to regulate the expression of GFP. So keep that in mind. So ARC protein sort of like an on-off switch per se. With the presence of arabinose, it can turn on the expression. If the arabinose isn't there, it can turn off the expression of GFP. So keep that in mind. So now, works as an on-off switch. right? So now earlier plasmids is exchanged with the conjugation in wild type E. coli. But we're not doing that. We want to put these plasmids into E. coli using transformation. So this is a transformation, right? So how do we do that is a question. Now, of course, when you think about it, plasmids are, it's a circular DNA. If it's a circular DNA, the, the backbone of the DNA is what? The backbone of the DNA is, it's a phosphodiester linkage, right? And so with, I mean, with the sugar or phosphates, right? So keep that in mind. It's pretty negatively charged because of phosphates. So it has, so in this diagram, you can see that, you know, it has lots of negative charge because of phosphodiester linkage, the phosphates. You have a lot of negative charge on the backbone. Of course, in vivo, you have magnesium to shield these negative charges. The same thing here, E. coli, it has glycolytic cell walls because it's prokaryotes. But remember, um, remember the E. coli also has a membrane, which is a it's a phosphate uh, uh, phospholipid bilayer, right? So which means it also has a negative charge, right? So there's an electric repulsion, electrostatic repulsion. So they don't really come too close to each other. Um, so what can we do is the question. Of course, you use calcium chloride. The bottle says transformation solution, but what it is just a calcium chloride solution. Of course, calcium being a cation is +2 charge, so it can actually shield these negative charges. So effectively bringing the plasmid closer to the E. coli than without calcium chloride. So once it comes closer, that's great, but it still doesn't go in. But remember, you're heat choking it. Remember, you're heat choking at 42 degrees for 50 seconds. So keep that in mind. Now, remember, this is 50 seconds exactly, not 51, not 49, but 50.0 seconds exactly. Because transformation efficiency is greatly affected. If it's 51, you may actually not as good of the transformation efficiency. If it's 49 seconds, it may not work as well either. So it's like be very diligent to get 50 seconds at 42 degrees Celsius. Now, at 42 degrees Celsius, it's enough and it provides enough energy um, uh, to go through the potential energy barrier. Of course, molecules are vibrating. So again, the 50 degrees, provides a pretty good vibrational energy for, um, the carbon and carbon and the carbon hydrogen stretching. But anyways, the point is that, it has it has enough energy to provide the potential energy barrier of this phospholipid membrane. And then when it comes closer, and then it has enough energy, and also you're putting it into the ice bath. When you put into the ice bath, the volume of the E. coli shrinks, and when the plasmid is closer, it sucks the uh, the plasmids that are in its proximity, and then transformation takes place. Of course, not all the E. coli can do that. Some of them may actually die out of the heat shock because 42 degrees Celsius is a pretty high temperature for E. coli. Um, but also, so you're quenching the heat by providing the E. coli into the cold temperature, in this case, the ice bath, and then it sucks the plasmids in, it quenches the heat. So, and after that, you allow the E. coli to recover because it's a pretty stressful environment for microbes. Cold, hot, cold, and then so you're gonna add LB broth just so that E. coli has chance for those surviving E. coli you hope for the best that they actually took you up that they uptook the plasmids, and then you allow it to replicate and multiply. All right, so again, point of this lecture, a couple of things. Again, you are in that's transforming, but this particular pGLO vector contains three regions. These three regions were replaced with GFP. And then Rabinos will make it as an on-off switch. This is important because if you don't have any, if your goal is not just to produce GFP, you also want to have E. coli that you want to grow E. coli that contains this particular trait. You don't want random microbes popping up in your plates. So we actually put ampicillin, but then if you have ampicillin, then generally, bacteria can't make it. But in this particular case, the pGLO actually has antibiotic resistance gene called BLA. So if the transformation takes place, whether it produces GFP or not, depending on the presence of arabinose, it can still make it in the ampicillin environment. So keep that in mind. And then, so again, so we make that happen by transformation process. So keep that in mind. So, that's it for the theory part. And the next day after the lecture, you'll be coming after about 24 hours. And of course, you want to write down the starting time and then ending time, because bacterial growth is exponential. Exponential. So, if you are trying to have someone else to reproduce your data, and then of course, if you don't write it down how long it was, then the person might just wait for 26 hours, and then there's a huge difference in population between hour 24 and hour 26, let's say, because exponential growth. So keep that in mind. Or it may not be, I don't know, depending on how fast the E. coli grows. So anyways, the point is that keeping track of exact time, if you are actually calculating transformation efficiency, will be important. You're not doing that, so it's not end of the world if you didn't write it, but it's always a good idea to write down all the actual starting time and ending time. Now, and once you actually look at it, write down the time, and then make observation and on 4 plates, and let's make let's parafilm it and put it into the fridge so that we can use a few of the colonies for next week's experiment. Speaking of which, week after that, not week after, but a few days after that, about a day or two before the subsequent lab, we're going to have to start with a liquid medium or media, because you're going to have 2 tubes. We're going to actually pick a colony from the one of the LB amp and AR plate or so, and then put into the liquid medium. So there will be an instruction because we'll be doing a protein isolation lab, which I will put up another video when the time comes. But protein isolation lab, we need the protein. The protein will be GFP, but you need to have lots and lots of GFP. So you want to give yourself minimum 24 hours, preferably a little bit longer than that. So somewhere between 24 to 48 hours prior to the next week's lab, you want to come and start the liquid culture medium. Instruction will be provided. So okay, thanks for watching, and I'll see you shortly. All right, thank you.